this type of analysis in biology is typically done by the fluorescence microscopy, which is, I would say, a working horse right now in many laboratories. And it's done because it is possible to tag a gene by molecular targeting, by attaching the GFP protein or any other fluorescence protein to the gene. For example, here on the right, you can see an example that ATO7 gene is involved in eye development, was basically tagged in this fish. Here you can see it as a panel below in the eye region here. One can perform this targeting with the antibody staining, also called in situ hybridization, where specific arrangement of molecules is targeted, right? So by means of fluorescence imaging, one can actually not just see the genome as a whole, but at the specific regions where it is at the moment expressed and acts on the structure and structural differences. So this location of the molecules, which is available with fluorescent microscopy, is possible to visualize in 3D by the light sheet microscopy, and this is the Bruker system comes in. So we have on the right, you can see here, a photograph of a multiview light sheet microscope built by Alexander Bruker. And the principle of it is very easy. On top, you can see here there are two cameras that are located opposite to each other, and there is orthogonal to that, there are two light sources actually coming in. So there is a light sheet created by the two lasers, and at the same time, one detects from two different directions. So independent of the type of a targeting, whether it's a genetic modification, antibodies, and situ hybridization, one could put a specimen inside, create a light sheet, detect from two different angles, and then rotate the specimen and basically detect several different volumes. And it's extremely helpful for the large biological specimens, like fish embryos, for example. They're typically only one part specific of the embryo would be sharp and close to the detection plane, while the other one would be more or less kind of blurred and out of the, not out of focus, but the signal there is not as sharp as possible. And so by creating like 4 different orthogonal volumes, one can merge it to the more or less isotropically a spatial resolution, quite large imaging volume, information on there the gene is expressed. And if one does it on the, in this case, this is a fish organoid, which we have done, but it's the same gist. You can see that ATO7 is a reporter for the eye cells involved in the retinal development. You can basically reconstruct a whole 3D volume of where this gene is positioned and which kind of cells and how does it act on it. What is interesting, however, often what one sees in fluorescent microscopy, one have an impression of a complete tissue, so to speak, and one see those individual cells, but there is a lot of information around which is kind of not present there. And that's because fluorescence microscopy is relying on the targeting. And my favorite comparison in this case, if one would like to compare fluorescence and X-ray microscopy, fluorescence microscopy is like seeing, in this case, it is San Francisco during the night, and the X-rays, it's like looking at the same town and during the day. So the X-rays provide you full structural information. There is no, however, specificity to any molecules, but we can see here every building in the town. You know, we can have a look at its height, we can analyze it, density, and etc. and so on. On another side, in fluorescent microscopy, there are always like those sparse dots, and if one does, for example, cell tracking, one has a feeling that the cells are propagating like in space and there is nothing around. So we can actually not see all the information, but we for sure know by this fluorescent targeting, I don't know, that in this specific window here, gene A or gene B is active. And that information is extremely crucial and important. And the correlation of both, not just knowing that, okay, this gene A is expressed, but it is expressed in these particular tissues within this organism, and etc., is extremely important at all different hierarchical scales. And this problem is very well realized and acknowledged on the level of a cellular biology, such that there is already techniques created to it. And one, I think, of the most famous is Clemet's correlated light and electron microscopy. So you can see here a correlation on the cell of the electron microscopy signal plus the nucleus label, and this is a Golgi apparatus inside, which is provided by the fluorescent microscopy. We can do very similar analysis on correlation of light And in this case, X-ray microscopy, this is a human osteoblast cell. This is the one which is forming the bone. And we can localize very nicely the protein, which is called G3BP1 protein, important in stress granules production inside the cells, even though structurally you cannot see what it's acting basically there. What is interesting that at the organismal level, while I think this problem is also quite realized, it was up to now typically solved by the use of a so-called optical tomography. These are the two, I think, most prominent examples. On the right, you can see that these blue structures, these are actually the lenses comes from just simple absorption in the same light sheet microscope as have been presented formally. And then the group of Jan Husken actually have developed such that you can do an acquisition, so to speak, in the spiral and then reconstruct in this case, it's zebrafish embryo, I believe. You can reconstruct basically a structure of the embryo also in 3D, and then you can localize the light sheet fluorescent signal inside. You can see, however, that from the composition, one can guess like the main parts of it. So I know that these are, for example, otoliths, these are the eyes, this is the spinal cord, but the information on the structure is not, so to speak, as on the histology level as X-ray tomography can provide. And so we wondered whether we can extend the light sheet microscopy imaging to provide, to combine it with X-ray tomography and provide more cellular-like tissue anatomy. And I think what is important here to say, but it actually makes a difference in which order to make the imaging. It's not just kind of, okay, I go either to that one. But we do first light sheet microscopy, when would take the same specimen, and then we would go through the dehydration and the staining with the contrast agents to create enough absorption for X-rays. And then we would do X-ray tomography with this high throughput system. So it is important to do fluorescent microscopy first before the X-rays are done. And what was also important for us that typically the specimens for light sheet microscopy are embedded in the low melting agarose, and because we want to go through more specimens and not try to actually then unmount or take out those specimens individually for the X-ray tomography scan, we actually instead of mounting them in agarose tube, we just put it basically on the agarose pillow. This is something what we have used before for the in vivo imaging. They then image it with light sheet microscope, and we'll just kind of pour it out the specimens. And from that moment on, they can just go through the staining procedure and then go to the normal epitube and basically go for X-ray tomography. That kind of handling the order and how we prepared the specimens allowed us to go through many specimens at the same time. And you can see here an example of the light sheet microscopy data on different types of specimens. There are 3 channels all the time. And you can, I mean, if afterwards one would look at the X-ray microscopy data, this would be looking like that. Okay. And so at this particular moment, you would need to believe me that this embryo here on top left is actually the same as the embryo here, because this difference in the contrast, like I showed you on the CT, comes into play. The contrast is completely different on one side and then on our side, because these are always 2 different microscopes. The orientation of a specimen is changing. There is a difference in resolution. So actually the main challenge afterwards comes to actually how to put this data back together. And for that, we have been working on the correlation pipeline. And I should say that even think about 3-dimensional data and also light sheet microscopy data, I think it's clear to everybody that typically we're working with very large data sets. And to simplify the computation, we have been working on first compression of a data set. So in the light sheet microscopy, you typically get many channels. And so instead of trying to work with each of them individually, we said, okay, the first task is to find the transformation matrix and then to apply it to the regional data. And for finding a transformation matrix, how to combine those 2 modalities together, one does not need